Today, our speaker is Dr. Anup Parikh. He is the Director of Product Marketing at 10x Genomics. I'll let Anup take it from here.
Great. Thank you very much. Can you hear me?
Yeah, I hear you loud and clear.
Okay. Perfect. Great. Well, thank you very much for the opportunity to speak today. Today I'm going to really focus on, as the title suggests, our solution for de novo assembly. But before I start into that, I just wanted to give everybody a bit of reference as to who we are as a company and what we do. So, yeah. So really, what we're trying to do is build an intersection between our capabilities as an instrumentation company, as a reagent company, and our expertise in software to build solutions across the genomics space for everything from whole genome discovery to single cell analysis, as well as our de novo assembly and targeted solutions. What we really strive to do is to bring people from different disciplines together within our group. And this is a picture, I think, from May of this year, where we've got expertise in computational biology, in chemistry, biochemistry, enzymatics, as well as a lot of what we call true software development. Folks from, we're based out of San Francisco, so folks from places like Google and Facebook coming and working with us to really scale software to try to tackle some of the big problems in biology that are rivaling the type of problems that we're dealing with on some of the tech companies here in San Francisco. So we've been around for about 2 years now, public for about 2 years now, and we've got 4 key solutions that we really focus on. We have a single cell solution where we look at doing cell-to-cell RNA transcription detection and doing it without having to average over the bulk of the cells. We also have a whole genome solution where we really try to use, and I'll talk more about this in a bit, the power of what we call linked reads to really fully resolve the haplotypes and the structural variation across the whole genome. And we can do this because we don't have to average across the chromosomes. We can actually split out the 2 separate chromosomes in the human at least. And look at each one individually so that, again, we're not averaging across. That same linked read data type is what we're using to develop our de novo solution, all the way from sample to answer, and I'll talk a lot more about that today. And then finally, our targeted solution, which essentially allows you to take the power of those linked reads that you would apply to the whole genome scale and do everything that you could do on the genome scale, but do it in a targeted fashion. So you're only looking at certain portions of the genome, but still maintaining the long-range information to resolve haplotypes, structural variation, and get into regions of the genome that previously were inaccessible. So these are really the 4 main solutions. And I say solutions because, again, we'd like to bring together everything from our upfront workflow for sample preparation all the way through to robust analysis solutions. And on the de novo solution today, you'll see how a lot of our focus has gone into developing software to do de novo assembly using the power of linked reads and really get to a type of assembly that really we've rarely looked at before. So one of the fundamentals that we like to kind of start out with here is to kind of remind a lot of the geneticists in the room that we are in fact diploid. And I'm going to talk about humans a lot for a large portion of this talk before I go into the other organisms that we've looked at, because a lot of our focus has been on human assembly and human analysis. But we are diploid, and therefore we have 2 chromosomes, one from the mother, one from the father. And typically what we've done, given the technology that we've had in hand over the last 30+ years, is to average across these 2 chromosomes and take a look at the genome as a haploid view. This is generally how we look at the genome view. And the reason we did this was for actually fairly good reasons. We thought that the differences between the 2 chromosomes were minor enough that we could just represent it as a haploid and then just look at the delta between this haploid view and what was different in the other chromosome. And that was great, and I think we learned quite a bit from that. But I think as we go further and dig deeper into the biology here, we're starting to realize that there's a lot of differences between the two chromosomes, and in fact, a lot of the science that we'd like to do is starting to become limited because of this haploid view. This is exactly the philosophy that we applied to our de novo assembler. When we started looking at de novo assembly, we again looked at the fact that there were multiple chromosomes that you were looking at, and just averaging across them, or actually just compressing them down into a consensus sequence. So how do we get to a true diploid representation of the human genome is really what we wanted to do with our de novo assembler. And that was where we first started before we started branching into looking at other organisms beyond human. So looking at the two haplotypes in human is an interesting exercise and requires some new tools really to be able to do that. The way we do this is really to look at heterozygous mutations across any part of the genome and say, can we find can we split apart the two haplotypes using these heterozygous mutations that are peppered across the genome. Now, a typical heterozygous site within the human is about it's about 1.2 kb apart within the human, at least, and this varies from organism to organism. Now, looking at our standard short read technology that we're all kind of used to looking at, this becomes really hard to resolve across, because short reads, typically 150 bases, maybe up to 300 bases, can't actually span across this 1.2 kb and say, can you disambiguate the paternal from the maternal haplotype? Long reads help a little bit more, but every about 200 kb, you get separation in heterozygous mutations that's greater than 20 kb. So even when you're looking at long reads, unless you can span across 20 kb reliably, you start getting into problems there as well. So it gets a little bit better with longer reads, but really to try to get true separation of the haplotypes, you need to get to much longer range information that's greater than 20 kb so that you can do this reliably across a full genome. And this is where the power of linked reads come in. So typically we're used to thinking about the short reads coming from various parts of the genome. So you can imagine each one of these dots is a short read, 150 bases or so, and really not organized. So what we'd have to do is map this back to a reference genome and look at where they are. What we're doing is actually linking all of these, hence we come up with the name linked reads. So what we do is actually take all of these 150 base pair short reads, and we can map them back to the original molecule from which they came. So what you see in the bottom half of the representation here is a line that represents the original molecule, and then the reads are the dots coming on top of that. So we were able to link all of the reads that came from one molecule together and come up with the linked read concept. So what this allows us to do is an interesting distinction that we can start making is we don't cover every single molecule in saturation so that we get a full reconstruction of that original molecule that you started with. In fact, what we do is pepper that molecule with a few short reads coming from it. So we can maintain that kind of 30x average coverage that you would look at a whole genome for human assembly. But really what we get is 150x of physical coverage. So we have a lot more molecules that are these long molecules that have peppered reads on them that we've actually identified each short read molecule coming, or short read coming from that molecule. So you get much more physical coverage versus the actual sequencing coverage that you do, the read coverage that you get. And this physical coverage from 100 plus kb molecules is what really gives us the power to, in the linked reads, to be able to resolve the haplotypes and gives us ability to do all sorts of things that with short reads we would have to either compute with using computational tools or just physically wouldn't be able to do. So how do we actually do this? Well, what we do is start with about 100 kb long molecules, and we put them through a microfluidic system. So you can imagine the schematic here. You've got a microfluidic system where each one of the dots here is a unique molecule, or sorry, a unique barcode of a different color, and we've got about 4 million unique barcodes. That barcode is actually put into an emulsion droplet with a molecule of DNA. And now every single droplet goes through an amplification where you create short fragments that can be sequenced, but every single short fragment is tagged with the unique barcode that was part of that droplet. So you can imagine each one of these colors being a unique barcode, and now the molecule that came from that droplet is colored with that barcode. So when we get to the bioinformatics on the other end, we can say that every single short read that came from a particular barcode came from an individual droplet and therefore came from the same molecule, and now we can start reconstructing that linked read map that you saw in the previous slide. And this is what gives us the long-range information while still only doing short read sequencing. So there's a lot of things that it allows you to do. One of the most common reasons why you want long-range information is so that you can span across highly repetitive elements or prologous elements where mapping would typically be hard to do. So if you can imagine a cartoon here where you've got two regions that are highly similar to each other, you've got some differentiated parts anchoring outside of these similar regions. You can map 150 base pair reads to those differentiated regions outside, but in the middle, you kind of get a mix of reads just mapping to either one of those locations, and typically what you get from your output is a low mapping quality score associated with it, and generally all the downstream tools will ignore those low mapped short reads because you can't ambiguously or unambiguously identify exactly where this short read was coming from. Now with linked reads, because you maintain that long-range information, once you've anchored on those outside regions, now you know that every single molecule that came from either the purple or the blue barcode or the red barcode go together. You can start putting these things unambiguously within these highly prologous regions and correctly map them. So this is where we get our ability to go across highly similar regions, but uniquely be able to assign where each one of the reads came from. So this is one of the main values or main attributes of our system that our assembler, our supernova assembler takes advantage of, is where there would be typically gaps because we couldn't unambiguously map to a location, we can now fill in those gaps because of this long-range information and being able to anchor outside of those repeat elements and still be able to uniquely assign a good high-quality mapping score to each one of those short reads. The second thing we do, which is what I originally started with, is being able to separate out the haplotypes. So typically you start out with two different haplotypes. You kind of shear the DNA, so you've got a population of haplotype 1 and population from haplotype 2. You map all of those, or you kind of compress all of those together into a consensus sequence. This is what we typically would have done because we couldn't disambiguate the different haplotypes. Now, what we do with our assembler is actually, because we can maintain the long-range information and link each one of those heterozygous mutations across a long stretch of DNA, we can start pulling apart the haplotype 1 versus haplotype 2 and look at it as two unique representations of a sequence rather than a compression into one consensus. So the way we do this really is we look at all the heterozygous mutations across a region and start forming two separate tracks of sequence, and we can say one came from the maternal DNA, the other came from the paternal DNA. And we just look at all the heterozygous mutations and any given location. And we can do this across megabase ranges, and you'll see some of that data later. So when you think about accuracy of assembly, and now you think about accuracy in the context of having two different chromosomes and not wanting to do that consensus that's compressed from the two different chromosomes, you can say, let's assume you have the mother and the father. Father is represented as a 0, mother is represented as a 1. Everywhere there is a sequence that you have in the child, you can start looking at heterozygous mutations and say, did that come from the father or did it come from the mother? And start creating a sequence where you have a series of 0s or a series of 1s, where depending on which original parental chromosome it came from. Now, in real biology, you'd have two separate chromosomes, so you'd have two separate sequences where everything would be 0s or everything would be 1s, because that's the two chromosomes that you originally are comprised of. So when you look at this across regions of the human genome, where we know, but where we have good consensus of unique sequence from a single human source rather than a mix of multiple sources, what we typically see is, in this case, we're looking at the kind of the HapMap samples, the Ashkenazi trio samples that we're all kind of familiar with in the human space. You see this kind of what our scientists like to call a Franken Pig, which is a combination of two different chromosomes kind of interspersed with each other. When we start doing this with our supernova assembler, what we start seeing is two different assemblies, one completely coming from the father. We definitely have some switch errors, and this is where continuing efforts are to improve on our software. And then one coming from the mother, where you have a contiguous sequence and looks much closer to true biology that you would have in every single cell versus the artificial chromosomes and sequence that we've been generating so far with the tools previous to 10x. So that's really the second power of the supernova solution, which is to utilize those linked reads and to start separating out the two different haplotypes based on the heterozygous mutations that are throughout any genome. And so when we put all of this together and we start looking at kind of the standard metrics that everybody judges de novo assembly by, you've got the contigs, which is the ungapped sequence. How good is the accuracy, is the perfect stretch across that sequence? And this is something that we've actually found difficult to do with our tool, because finding a perfect stretch of sequence requires you now to get to the haplotype view of every single sequence. If you're switching between the maternal and the paternal sequence in your reference, then you can't really assess accuracy anymore, because you don't know whether, because our tool is starting to actually give you unique maternal sequence or unique paternal sequence, and there might be differences. Scaffolds we kind of know about, and then the new thing that we're starting to put within our output is to look at the phase blocks, where we can actually move this kind of consensus sequence into its unique haplotypes. And we're starting to see typical phase blocks of 3+ megabases, and a long stretch of phase block is upwards of 10+ megabases of separation that we can get. So this is really the type of output that we're starting to get to, is looking at per haplotype view of diploid assembly. So when we do this on the human genome, we looked at NA12878, which is a standard reference material that I think most of the human community is working with. Start with about 1.25 nanograms of DNA, which is, again, critical for us, because we don't want to start with lots of material, especially when you're working with smaller organisms. You end up with, if you needed to get nanograms or micrograms of material, you might have to get multiple organisms kind of averaged together to get enough DNA source to actually do this. So our ability to get down to very small amounts of input material allows us, especially for those small organisms, to start with one unique individual organism and do a de novo assembly of that genome rather than an aggregate of multiple organisms, simply because of the DNA input requirements. In this case, we had an average molecule size of about 80 kb. And we use the standard Illumina HiSeq sequencer. So this is where, again, we can take advantage of these fleet of short-read sequencers that are out there that are very cost-effective to get to very high throughput sequencing and do it in a rapid manner. We've developed our supernova compute requirements to, again, try to do this in a much more routine way, so you don't need a lot of extra horsepower to do this. We were able to run this on a server that had a fairly high memory requirement at the time. We brought the memory requirement down to about 384 gigabytes now. We're able to do this in about 2 days with a fairly standard compute environment. And with that, we're able to assemble the whole human genome at 56x coverage into about 1,600 scaffolds. Our N50 config length is about 100 KB. Our scaffold size is about 14 megabases. And the last kind of metric that's really interesting is, in this case, our N50 phase block was about 2 megabases. We've actually made improvements on there in the latest software, and we're getting over 3 megabases of N50 phase blocks now. All of this data is publicly available to all of you. It's available on our website. It has all of these metrics associated with it, as well as the software if you'd like to go and do your own assembly. Everything is open source and available to you. So in terms of the software, as I just mentioned, we try really hard to try to make software that is consumer friendly. Since we have a great team of people that are of the mindset that they would have worked at places like Google and Facebook, we think about software as being something that should be easy to use. It should be run anywhere that you want it to be run and not require a lot of extra work on your part to do. So the package of software that we provide for this assembly works on a standard standalone system, requires fairly decent amounts of compute and 24 cores and about 384 gigs of RAM. The whole thing takes about 2 days to run with a standard human genome, and the software is, as you can see at the bottom there, with those 4 commands, you can get the software up and running as long as you have a standard Linux system available. The whole thing is self-enclosed. There aren't any other dependencies, no need to compile anything. Simply download and start running with your short reads. And we have this philosophy for all of our other software for genome and single-cell analysis. It should be very simple to download and start using, and data sets are available as well as the software on our website. So beyond just the work that we've done in human, when we started this assembly project, product development, we have lots of people interested in sequencing and doing de novo assembly of a lot of different organisms. So we very quickly started getting requests and bringing in sample types from all sorts of different species to see if we could do assembly with a tool that we had originally designed for human de novo assembly. So I'll put up a bunch of caveats in terms of what we think we can do versus things that we haven't tested and some recommendations. These are just a distilled set of recommendations. We have a long list of guidance on our support website as to what we think is doable versus what we think is not doable versus things that we just haven't tested. So the first thing is, really, we think genomes between 1 gigabase and 3.2 gigabases in size are ones that we can support. Getting bigger than 3 gigabases, I think we start running into compute challenges. Going lower than 1 gigabase, we start getting into challenges around how our linked reads work and how many partitions we have. Smaller genomes don't behave so well with our linked reads. It's definitely a limit that we're starting to push even lower, but this is what we feel comfortable with today. Again, we built this tool for human assembly, so it's been well tested on diploid genomes, mainly human. We haven't tested it on haploid, but given the way it works, it'll just collapse everything down to that haploid view because it won't find any heterozygous mutation. So we think it should work well on haploid, but we haven't tested it very much. Polyploid, definitely not something we've tested and probably will require some improvements to the software to be able to support. So that's one where we really don't have a good answer in terms of how you would go about doing it. We definitely think that DNA should be obtained from an individual organism, because you don't want to get into this problem of getting into this Franken Pig, as we like to call it, from multiple individual organisms, or from mixing of chromosomes. Most of our testing, just given the wide variety of genomes that are out there and the type of complexities that different genomes have, we haven't really tested that whole dynamic range. So we think genomes with repeat content that's similar to human and GC content that's similar to human should behave fairly well. Once you start getting to the extremes, we really haven't tested it, so we don't know how well it would work. It may work, but we just don't know. Lots more information available on our support website around what we think will work and what we think may be difficult. So this is just a distillation of what we think are genomes that we could probably handle as the software is today, or as the pipeline is today. So we have tested a bunch of genomes. Like I said, we got lots of interest from a variety of different groups wanting to look at assembly. So we worked with many human-specific samples. We also looked at a lot of non-human samples. Komodo dragon, spotted owl, hummingbird. And you can see in the first 4 columns, these are the kind of the standard things that everybody would look at. What is the size of the genome, the size of the DNA that was input into the system, and that ranged based on how people were doing DNA extraction. So that's definitely one that we have lots of recommendations on our website. We want to get to the biggest size DNA as possible to maintain that long-range information and to truly be able to do the assembly and phase everything. Our N50 contigs are around 100 KB, and our scaffolds are about 10 megabases in this case. What's on the last 2 columns is really where we start looking at our ability to get to the individual haplotypes, given that we've got this really long-range information. Now, the reason we put up the heterozygous SNP spacing is because if you've got a highly inbred population, you can't get a very big phase block. So when you look at something like the Komodo dragon, which is a highly inbred, your heterozygous SNP spacing is 10 KB apart. This is why our phase blocks are so small, is because you don't have a lot of variability across the 2 chromosomes. Whereas things that are fairly well not inbred, something like the hummingbird, you get really nice phase blocks across that entire genome. So these are all collaborations that we've done with various people across the country. All of them have this data within some pipeline for publication or making it public to the rest of the community. So we're just showing kind of the high-level summary metrics here, but clearly there's more data behind each one of these rows, and it will be coming out through the collaborations that we've been doing. We were able to look at one specific human sample from the Human Genome Project, where we looked at about 300 megabase region, where we knew that that whole region came from a single person, and that it was phased so that we knew exactly which genome, exactly which haplotype came from which maternal or paternal chromosome, and therefore we could actually measure the N50 perfect stretch. How much of the sequence did we get accurately? And we were able to get about 19 KB of perfect stretch. This is really hard to do because we have to be able to find a truth set where we can get to the individual haplotypes. And so it wasn't done for every other organism that you have on this list because we don't have that kind of a truth set. But given the accuracy of the Illumina platforms, we're pretty confident that we can get to highly accurate contigs once that assembly is done. So with that, I'll just kind of summarize our overall workflow. We've got an end-to-end solution where we think within about 2 weeks, or a little bit less than 2 weeks, we can get to full assembly of a diploid human organism, human assembly. DNA extraction, given all the different types of species that are out there, will vary based on how you're doing the extraction. Could take, gel plugs giving you the highest quality DNA, taking quite a bit of time, to standard extraction from blood or cells taking you a matter of hours. We'd like to get DNA that's greater than 100 kb, but we can probably accept things that are 50 kb or higher. The metrics that I showed in the previous table will definitely suffer as the DNA length is shorter. Once you have that DNA extracted, hopefully high quality, long length DNA, you're basically just creating a single library, and you're doing that from 1 nanogram of DNA. And then you put it on a standard Illumina sequencer. A lot of the large genomes we've been using, our collaborators have been using the HiSeq X systems with the standard 2x150 reads to get kind of maximum throughput at a quite reasonable cost. And then we have our de-novo assembler tool, the Supernova assembly tool, which is again available on our website, which runs on a standard system, and will start outputting diploid assemblies as we talked about, and has been tested on various other species. So this is really the solution that we're striving to deliver. We've got a version of this out there. We've already got a rev of that version that went out in September. So we're rapidly improving on the software that we have out there as we get more feedback and look at different organisms and kind of start seeing the edge effects that are happening. So this is an active development for us, but definitely a fairly stable tool available today for all of you to be able to try out. So with that, I will stop and take any questions from all of you.
Hi, so this is just one more reminder that please email or your questions to anna.childers@ars.uscu.gov or send a private chat to Anna for any questions that you may have. So I actually, we have one initial question here. Do researchers need to plan to use 10x genomics approaches from the start of the project, or is this data easy to generate and incorporate after some preliminary sequence has been generated?
What we found is DNA quality matters a lot. So depending on how the DNA was extracted, you could have some challenges if you're in the middle of a project. Longer DNA is always better, and we've seen performance we've seen the best performance when you have greater than 75 kb, 100 kb molecules. So that could be one issue, but otherwise we have seen customers looking at ongoing projects where they might have previous Illumina data, previous PacBio data, or even BioNano, and looking at how they can put these things together. Our solution won't put all of the various different other tools together to make the assembly, but we definitely have, actually, there was a recent publication on human genome from Korea where they use multiple technologies to get the best assembly possible. So it's definitely doable.
Great, thank you. I next have a question about, so do you foresee using 10x for metagenomic assembly?
Yeah, we have had quite a bit of interest around that. We think it's kind of an ideal fit in looking at the phasing as well as getting the deep counts that you need for metagenomics. We haven't yet developed any software to do interpretation on that, but we think it's a really ideal application for it. So I think at the simplest, I would say the answer is, yeah, we definitely see use of 10x in metagenomics, but we haven't developed a solution around it yet.
Okay. The next one kind of dovetails with that, is about contamination. Do the microbe DNA, do they just fall out of the analysis?
We haven't looked at metagenomics type of applications. Generally, we've been working with pure DNA, so I couldn't say exactly how contamination would be affected in our solution with the standard solution that we have. We have had customers basically take, when you're doing the reference-based analysis, take our reference assembly and put in new contigs or new fake chromosomes for other species to try to pull out things that might be incorporated in the human genome, so viral incorporations and things like that. So I have seen things like that fall out quite readily when you do reference-based analysis. I don't have very good data on the de novo assembly part of it, so I can take that as a ask and see if I can get some answers on that.
Okay, great. Next is, do you see any differences in performance in assembling heterochromatin versus euchromatin?
I don't think we've looked at that very specifically, or at least I haven't seen the data. Maybe some of our scientists have. I think we're going to have to go back and look at that. I don't have a good answer for you right now.
Okay. Next, does 10x Genomics publish a catalog of core facilities with a machine that are able to provide the service?
We have not published anything as such. We have a couple of agreements with service providers that will do that, so we have those in our kind of corporate news and view section. Probably the best way, though, is we're way past those sites. We have lots more sites that can do this. So the best way to do this would be to just contact us through our website or shoot me an email, and I can put you in touch with your kind of the regional salesperson, and they have a list of places that have an instrument and can do the type of work that you might be interested in.
Okay. Our next question is, can the library prep method be used along with a long-read technology in order to increase contig sizes?
Yeah, it's an interesting question. We've definitely had customers be interested in that. We have right now developed our entire solution around the Illumina sequencers, so we haven't tested that yet. I know that there are people that are interested in doing this and some that might be trying it, but we don't have very good data and a good feel ourselves as to how this would work and how complicated it would be.
Great. So just one more reminder, if anybody else has any questions, we're welcome to take them at this time. So Anup, while we're waiting for people to ask stuff, do you have any other take-homes or points that you'd like to bring up at this time?
Well, I think, I can always keep talking about our technology and kind of what's going on there. I think one of the more interesting things around de novo assembly that we're starting to look at is how we could augment our kind of reference-based analysis that we do today for looking at short variants of SNVs and indels and such, but use de novo assembly to try to improve in areas where we know we have high polymorphism, so reference-based analysis doesn't work so well, or areas where mapping back to the reference, especially with things like insertions and deletions that are larger, it's hard oftentimes to do that mapping back to the reference. How we can augment reference-based analysis by doing some of this de novo assembly in parallel. So that's another area that I didn't talk about today, but it's definitely one that we're working on to look at how we could improve, how we could use the best tools possible. Reference-based can do some things, de novo can do other things. How can we use both the tools together to get the best possible view of a genome?
Well, that's actually a really interesting point that you're bringing up, the idea of being able to improve existing assemblies with this hybrid approach. And you said that's something that's actively being worked on. Do you guys have any sort of GitHub or public repository where people can actually check out your open source projects that you're actively working on?
Yeah, so I'd say there are two ways to get involved here. One is we've got all of our software that I talked about today is available open source, so that's something you can check out, and we periodically update that as we incorporate new things. So this would be an area where we might incorporate new things over time. More immediately getting involved would be we have a 10x community that is associated with our website, which is an open forum where we're starting to develop kind of virtual communities where these types of discussions can happen and can be facilitated through our R&D organization so that we can put out data sets, put out snippets of code and try to do some of that. And de novo assembly and reference analysis or reference assembly is definitely an area where we're going to actively create a community and try to build some of that interaction amongst everybody else in the field. So it's definitely a website worth checking out. It's community at 10xgenomics.com.
Great. I have one question here. How does the amount of Illumina sequencing for this method compare to standard de novo Illumina sequencing?
So we right now recommend about 56x coverage on the whole genome. Reference-based analysis, the community generally does about 30x coverage, so we need about a little bit less than twice as much coverage to try to get the best de novo assembly. We have looked at kind of subsampling that, and we feel that a lot of our assembly metrics are quite well maintained even at lower than 56x coverage. But given this is first iteration of our tool in the last 6 months, we're still recommending 56x coverage. But we think we can go lower than that and still maintain a lot of our N50 numbers. I would say standard short reads just alone, there's just the long-range information that you need. No matter how much depth of sequencing you do, you're not going to get a lot of that. So your N50 phase blocks or N50 contig lengths or scaffold lengths are just not going to get there with longer or deeper coverage. You're going to have to use some sort of long-range information to be able to put those two together.
Okay. Will longer Illumina read links from the MySeq also work with the current assembler?
So right now our assembler is quite well optimized to the 150 bases. We think that maybe long range or long reads might help a little bit. We haven't done very much testing on that, so it's kind of hard to say, because most of what we focused on is larger genomes, and there using the HiSeq fleet is what we've really worked on. And so 150 base pairs is really what we have access to, paired end 150 bases.
Okay, thank you. So if nobody else has any questions, I'd just like to go ahead and I guess we can wrap this up. Thank you everyone that called in, and thank you again, Anup, for such a great presentation and this fascinating discussion. Our next webinar will be on December 7th, and that will be Dr. Ivan Lyachko from Phase Genomics. He'll be here to discuss the HiSeq chromatin interaction mapping method and its applications for chromosome-level genome scaffolding and metagenomic deconvolution. And I'd like to just wrap with just a final reminder that we will be posting links to both the slides and the recording of this presentation later this week. So keep an eye on our website and the YouTube channel for more information about future presentations. Thank you, Anup.
Great. Thank you, everyone.